Those that are on the webinar today, thank you all for joining us to listen to the webinar. This webinar will deal with the automated platform for drought and multi-treatment experiments, but not in isolation. We will relate it to the Journal of Experimental Botany editorial paper that just went out fresh, which is called Guidelines for Designing and Interpreting Drought Experiments in Controlled Conditions. And so let's begin. A few notes just before. So this webinar is recorded. So please, if you can kindly keep your webcam and microphone off during the talk. And also, if you have questions during my talk, please raise questions in the chat. We will address them at the end of the session. So some background about us before we start diving into the topic of the webinar. So we are Plant-Ditech. We developed the functional phenotyping platform, which is measuring plants in high throughput. And this system is a spin-off from the Hebrew University in Israel, developed by two professors in plant physiology and soil and water sciences. The company is commercial already since 2017 and already have established customers in different places around the world. You can see some of the names, the distinguished customers here listed, and we have many more around the globe. I'm Keren Moshelion. I am the CEO and partner at Plant-Ditech. I'm already with the company for 8 years since the start of its commercial phase. The agenda for this talk is to discuss about main pitfalls in drought experimental design, which were covered in the editorial from the Journal of Experimental Botany, and how PlantArray addresses these pitfalls. And we will also share different use cases for drought and multi-treatment trials that were done by our customers on the system, of course. At the end, we will have time for Q&A, so please, I invite you to write Q&A in the questions in the chat during my talk, and I will address them at the end of the session. So let's begin. So in the paper, in the editorial paper of the Journal of Experimental Botany, they covered several pitfalls, but I kind of put them into three different buckets that are the main buckets. One is to ignore, so I will just explain a little bit. The editors, what they did, they responded to many common submissions that were done to their paper, and covered these major pitfalls that they just came across when reviewing these papers. And they saw that there are some common lines between them. And I will cover the three main lines that relate to the experimental design. They also had some mention of other topics like definitions and terminology. I will not address this, but I will address whatever related to the experimental design. So these are three major topics. One is that a lot of the papers that are submitted, they ignore the G times E interdependencies. The second item is what actually to measure. And there are different traits that can be measured. Some are better, some are less. So we will cover that. And also the method to measure, how to measure. We will also discuss that. And the third point is about non-comparative drought conditions. So this is a major topic in the paper, where they cover a certain area, which is kind of forgotten in many of the papers that are submitted to them. It's not exactly forgotten, it's more of biased because it was not deeply thought about. So I will discuss it, here in the talk. Let's start with the first point, ignore G times E interdependencies, and why it's important not to ignore them. I guess for some of you it's very clear, but maybe so to others not. So the plant is being grown in his environment, and the environment is the atmosphere. It's the radiation, the temperature, the humidity, which makes the VPD, and soil, so different soil types. So the plant that is surrounding feels a certain environment of water content, of pH, of EC, of oxygen levels, of other characteristics. The shoots, of course, also react to the atmospheric conditions also, and it comes into play mainly in the stomata aperture, in the stomata activity, stomata conductance, and therefore how much CO2 actually comes in, how much water the plant transpires. And the root, which is also reacting to whatever it's in its environment. So the root conductance, water uptake, nutrients uptake, etc. And we have the rhizosphere with the all the activity that actually happened underground. And all of these elements, they're actually interdependent, because when there is a change in the environment, either within the day or between periods, the plant immediately responds. And also the soil characteristics around the plant is also changing, because if there is, for example, high VPD, of course there is higher also evaporation and also transpiration from the plant, etc. So it's actually a very interdependent activity that happens between the plant and its environment, and we cannot ignore it. And when we design for drought experiments, we need to take these into account. So as you saw before, we are talking mainly about the soil, the plant, the atmosphere. So the least that we can do is to measure this and not to oversee this and not to avoid looking at it. So this is the soil, the plant, and the atmosphere on a continual process, because during the day there are changes in the atmosphere, changes in the soil, and the plant is reacting. Also, what you could see in this is that the water is a very clear factor of the environment of the plant and the activity of the plant. So the water, either in the atmosphere, is very important, the humidity, or the soil water content is very important, the root water uptake, the shoot water loss, the activity of the water or the reaction to the water status is also very key when talking about environmental studies, especially drought. Also, what is very important is to make sure that what is measured, if it's the plant or the environment, is measured at the same time, because the environment is changing during the day, morning light, during the day, also humidity and so forth. So on an ongoing basis, to measure the plant and the environment at the same time points. This is very important as well. So this way, we can actually measure apples to apples and not pears to apples. So the PlantArray system takes all of this into account in terms of the soil plant atmosphere continuum. You can see here some example graphs coming from the system. What is also important to note that not only that the system measures the soil plant atmosphere continuum continuously over the day, but also in a randomized structure. A randomized structure which ensures that all the plants and all the different genetics, different treatments are done in more or less equal environment. So we discussed about the G times E and how we can take them into account if we measure them, right? By the way, I didn't mention, but the PlantArray system also normalized to the environment. So some of the parameters are being normalized to the VPD, for example. Again, in order to relate the plant to the environment. So let's assume that we already figured out this, and we measure now also the plant and the environment, the soil, the atmosphere all together. So what to measure? What are the traits that are key in order to understand the plant-environment interaction? So let's take this example of looking at the plant. A lot of what we do and a lot of what is being done in science is by sight. So if we look at the plant or measure the chlorophyll content or the leaf angle or any of that, let's look at what happens in drought. So in drought, we have here two examples. Now we apply drought conditions. Let's look at the upper picture and the lower picture. The upper picture, we can see a very vivid plant, which looks nice. And we could say, wow, this is a great drought-tolerant plant, right? It looks vivid, so in drought, it's really withstanding drought very nicely, where the bottom one actually looks very miserable. So we could come to conclusion that this upper one is drought-tolerant, but actually no, he is a drought survivor. He's closing the stomata in order to survive, in order to reserve better his water capacity or water within the plant, the reserves. Where in the lower one is actually could be a drought tolerant plant, which is taking more risk and releasing more water to the air. So can we really take conclusions based on sight only? Definitely no. What we need to do is to look at the plant activity, not just how the plant looks, because how the plant looks could be misleading, could be right, but it could be also misleading. So we need to take into account what is the activity of the plant, what is the physiological response of the plant. So physiology, when we talk about plant environment, takes a very central role in the process. Now, we saw before when we talked about the G times E interaction, we saw that the plant water status is very important because water is like the essence of the plant activity. Every time the plant reacts to the environment, it can close or open its stomata. So the stomata helps to define, the stomata aperture helps to define how much the plant is productive, how much the plant is taking risk, and how much water he actually loses, because every time he enters CO2, he also loses a lot of water. And how much does it affect the whole water flow and the water balance within the plant? So it has a very key role. And also what we know from the science, this is something that we all know, that transpiration, stomata, activity, all this, because of CO2, we know that it has direct correlation to yield. Also, the opposite of it, the plant water loss, has direct correlation to yield. So actually, if we can quantify the transpiration at the stomata aperture or conductance, we can envision or make a very nice comparative assessment which of the plants will have better potential for better yield, more yield potential. So just by looking at the water activity by the plant, again, flowing through the stomata. In our case, in the PlantArray system, we are looking at transpiration. We are also looking at root water flux. So we are actually looking at the whole process. From the root water uptake through the plant, we calculate how much water is within the plant and how much it is losing to the atmosphere. What you can see in this slide is how the system operates. On one hand, it applies the soil water conditions. For each and every plant, different cocktail at different conditions per the experimental design. And on the right hand, you can see the soil plant atmospheric sensing. In green is all the plant sensing, transpiration, biomass gain, stomatal conductance, transpiration, stomatal conductance, roots water uptake. All of these, we take about 500 times a day measurement. So this means continuous measurement all day long. Biomass gain, water use efficiency, this is taken once a day. And together at the same time point that we take the transpiration, stomatal conductance, and root influx, we are also sensing the environment. So we have the same measurement of plant and environment for about 500 times a day with the same time stamp, where we are sensing the barometric pressure, the humidity, the light, temperature, VPD, of course, it comes out, and additional environmental sensors like the wind, the soil sensing. These are extra that you may add if you want to. So soil sensing, we have a lot of offering there of different kind of soil sensing. And of course, one of the most important is the soil water content. And also reservoir sensing for those that deal more with the nutrients. All of this information goes into a cloud software where it's all being analyzed and compared with statistics in real time at the time of the experiment. So just think about the amount of data that you can see already analyzed while the experiment is going on. The technology is based on lysimeter, on scales. You can see here, just activate my laser. You can see here that we have a scale, mini scale. Underneath this gray cover, there is a scale. And what we are doing, we are learning with the help of the scale, the water status in the pot. And based on the fact that we are covering the pot from all angles, from the top, from the bottom, we can measure very precisely the water loss. So we are measuring the water loss, a direct measurement based on the mini scale, based on this very sensitive scale. And we also have atmospheric sensing together with these scales, and also you can add more plant sensors and soil sensors as well. So all of this, it's our development together with some additional parts that help to make sure that the precision is maintained. You can see here how the system works in actual. Probably I have to switch. Sorry. Sorry for that. I have to activate it again for the video to run. So you can see that the plants are growing on the bench. It's well irrigated, drought, and then recovery. All is actually seen in real time. You can see it by histograms. You can see it by heat map. You can see it also analyzed with box plot, with graphical view, with ANOVA analysis, t-test, and so forth. So what you see here, I will just mention this. You can see well irrigation, daily transpiration, 1 month trial of well irrigated, drought, and recovery, all within this 1 month, already analyzed statistically. And if you log in, let's say after 2 weeks, you will see the history already analyzed. So we discussed about the G times E interdependencies that are very important to capture and even to normalize. So whatever the device you're using, this is very important. The other thing is what to measure. We discussed about physiological traits and their importance. And also the randomization is very important. So there is averaging out the greenhouse effect. And there is another topic which is very, very critical that was emphasized in the editorial paper. It's about non-comparative drought conditions. Let's dive into that. Okay, so we have here like a common case of a drought study, which I'm sure you know. You see high transpiring plants versus lower transpiring plants, and they are being irrigated the first day, fully irrigated in the pot, and then we just let the plant to transpire. We don't irrigate again. So this is day 1, this day 2, day 3, day 4. Okay, so we see here 2 different lines of plants. One more plant transpiring, the other is lower transpiring. So based on that, what we can see is that after day 3, we can see that the lower transpiring plant is transpiring more. Also, is making more photosynthesis. So we could assume that this lower transpiring plant is a very good tolerant plant for drought. The question is if this is the right conclusion. Now let's have a look at another variable, which is the soil water content. What's happening in the soil? So if we compare these 2 lines, the first day, both are getting the same amount of water. So these 2 type of plants, they're getting full irrigation, no problem. But then what happens? The high transpiring plant is losing more and more water from the soil because it's transpiring. So he's actually pulling more water out of the soil and transpiring it to the air. The lower transpiring plant is going more gradually down, so less aggressive, and is also transpiring less. Because of that, probably this is what's happening, is pulling less water from the soil. So what do we see actually in these graphs? We see that the blue one, the high transpiring plant, came into a stress situation very early in the process. So day 3 already, he was under stress. But was he under stress because of the right, let's say, the right reason? Because what happened is it became in stress because he had no water availability anymore in day 4 in the soil. So in day 3 and 4, he already struggled, but he struggled because he didn't have enough water capacity in the soil, not because he is drought tolerant. And what happened in the lower transpiring plant? He was not in stress at all because he didn't feel at all that there is any limitation. He was not in stress, so he felt good and continued to produce. No problem there. So does it help us really to learn, does it help us really to learn if the plants are under drought tolerant? Is this really what help us to learn that? No, because the soil water content actually was affected. You remember that G times E. The plant is influencing the soil condition. So what do we need to do in order to really assess drought conditions? We need to provide them with the same soil water content condition. In this example, this is measured by the system. And this is just raw data of very initial, just raw data, not analyzed, in a very simple kind of drought experiment. I say kind of because we don't maintain here the soil water content. What you see here is just the response by the plant when we close the tab. And what you see here is a very big span of reactions by the different plants. These are different genetic tomato plants. And it's just closing the tab and letting the time do its work, but the soil water content is actually very different. And each plant has its own strategy. So we cannot really learn something valuable from it. What we need to do in order, and this is what also the editorial, this is what they recommend. What we need to do in order to learn the soil water content, the drought tolerance, how do we learn? We need to make sure that the soil water content is maintained the same for all the plants, all the time throughout the drought implementation. So how do we do this with our system? We actually use a feedback methodology with the help of our electronic card in our controller. This controller sits next to each and every of the pods. It can control two different solutions to be applied to the plant. You see tab A and tab B. This is the tabs that open and close the irrigation. And we can add different sensors here. So one of them could be soil sensor. So you can maintain the soil water condition based on the soil water content that you are sensing and provide the irrigation accordingly so you maintain the same soil water condition. We like this feature, which is by transpiration. We are already measuring the plant activity. So remember the interdisciplinary between the plant and the environment. We are already measuring the plant water loss. So we can utilize that in order to provide the same soil water content according to own plant activity by transpiration. How do we do it in practice? So with the help of this controller, please follow this line here, which is the soil water content. This is a normal drought experiment. In this example was done with the tomatoes. And you can see this is one line and we control this soil water content here. In the meantime, we also measure the plant. So this is daily transpiration in this example. Now, if I'm adding another group, you see that we continue to maintain the same soil water content. So you don't see what you saw here, that there is a big span. But here you see the lines going down more or less together. And we measure the plant activity while maintaining the same soil water content. This is key. This is really key in order not to bias the result. So what you see on the top is daily transpiration. Here you see two different types that react very differently to the same soil water content reduction. So now you can really learn that this plant in green is closing its stomata very fast and recovering very fast. So you can learn versus this red line. So this is, for example, now let's look at additional group, see their behavior, but still, don't forget, we maintain the soil water content the same. And now you can really look at the results and analyze the differences between the line. The system does it for you. This is the statistics coming from the system. So you already can see this and understand what your plants are doing while maintaining the soil water content. This is critical in order not to have biased results that you think that your plants are doing, having drought tolerance, but they're actually not because of the soil water wasn't maintained correctly. And it was another variable, another noise that came into your experiment. So with this system, you can reduce this to 0 and have just the soil water content controlled all along. Now, not only that, now we learn that the soil water content is critical. It's critical to maintain it in order to get to the right results or to the right understanding of the results. So what we did, we put the soil water content now in the x-axis. So you can see here that the soil water content on the x-axis and the traits that is being measured, the transpiration rate, is on the y-axis. Now we can see not only so in this when we do this, by the way, we can identify what is the stress point. This is the point where the transpiration rate starts to being reduced at this soil water content. So let's say point 2 here, this is the soil water content where the theta grid starts, and from that point on, the plant is in stress. Now you can see more lines together, and we can learn about the behavior of the different lines with their stress points, when they start, and how are the differences between them for each and every soil water content level. So this is another way to look at the data and learn about the differences between the lines, where the soil water content becomes to play a very important role in the understanding of the plant tolerance to drought. Another element which is also could be a limiting factor for pot trials is the pot size, because if we don't take care to provide the suitable pot size, then the plant can come to stress at the point that we didn't plan for it to come into stress. And so we I think I got disconnected. Okay, let me just do a check here. Linoy, can you confirm that you
Yes,
can
yes,
see me?
we can hear you and see you.
Thank you. So I'm bringing up again the presentation. Okay, so we stopped here. So what I was saying is that we can see that we have a solution for different plants with different sizes that suits with the same technology. You can fit whatever your plants are. Okay. So in kind of a summary at this point, what this system helps us to overcome all this biased information. So we measure the plant, we measure the environment, we measure randomized, we put it in randomized structure, we measure the physiology, we measure the environment at the same time, and we also control the soil water content in a very effective way. This helps us to shorten the whole cycle of the experiment. Now you don't need to wait until you get yield in your trial, because already within 1 month you can get a very clear understanding of well irrigated drought and recovery, what the potential yield of your plants are, and also how the plants behaved in each and every stage, because you can also see it in real time. So that it's all designed to shortcut the time and help you also to get much clearer understanding of how the plant are being responding to drought conditions or any other conditions that you can apply either through the solution or also outside. You consider the system can come in different sizes, from small few units to mid-size, mid-scale to large scale. It's all about budget and size, but we can fit every demand that you have. Another thing is that it is modular. So if you kick off with a certain structure, you can always add, you can add more sensors to it at any point of time. We almost didn't touch yet the continuous measurement. And beforehand, I would just say that one of the major problems for measuring continuously throughout the day is the signal to noise. because the environment poses a lot of challenges on the measurement. Every measurement, every sensor is actually exposed to the environmental influence. A lot of the effort went into understanding what can influence, what environmental phenomena influence these noises and take care of these noises in order to get very fine lines. As you can see here, these are graphs taken from our system, from our Spark Analytics, where you can see very nice, clean data, which was, by the way, compared to real stomata aperture opening. So the stomatal conductance measurements here were compared to, specifically in this trial and this paper that is just underneath, and was compared to real stomatal aperture, and you see a very nice correlation between the two. So you can check this out in this paper. By the way, all the papers, we have about almost 70 are published in our website. All these papers that we're using the system, you're able to log in to the website and see it under references and scientific articles. Here you can see, for example, how very nicely, you see this is a measurement of the environment, so light and VPD, and you see very nicely the correlation between stomatal conductance. You see this pattern, daily pattern. So this is on a momentarily, every 3 minutes we take a measurement, so throughout the day, and you can see how nicely it fits with the light, right? You see the light. So it has almost the same pattern, and transpiration fits well with the VPD. As well, you can see very nicely how it fits the pattern. But also you can see the differences between the line, which is the most important thing. So you can see which plant is reacting better than the others, like the blue one here, in a certain time of the day. And this certain time of the day, which could be only 2 hours, can impact the plant yield in about 15-20%. So it's quite dramatic. So as I said, we put a lot of effort to minimize the noise. There are many, many different elements. I will not detail them here. It's a patent technology, and every bit of the system was thoughtful, taking into account actually this element of the signal to noise. Thanks to that, we have very high correlation to field yield. There are papers about it. One of the papers took 10 years of phenotyping in the field, compared it to experiments on the system, and you can see that in well-irrigated terms, it's a 0.94 correlation. In stress, it's about around the 8.8. So it's very nice figures, and you can even see it by the eye, comparing the left-hand side, which is the field, to the right-hand side, which is the system. The blue is in well-irrigated, and the orange is in stress. I'll talk about some use cases to give you an idea what can be done with the system and how it can be done. So we discussed a lot about drought. So we have many, many papers dealing with drought. The system is really designed for it, for drought experiment, as you could see, with the soil water content control and also the continuous measurement and so forth. And you can see here a paper from China with cowpea and soybeans. We replicate 3 time points, 4 stages, and 2 species, where we measured the environment together with the plant, and you can see here the structure of the experiment. Using also the feedback irrigation, as we discussed before. This is all in the paper. Again, many paper can be found in our website. Another paper with barley from Göttingen. And here, what's nice to see is what we discussed before, how transpiration is correlated very nicely with yield. And you can see a live example from this paper. But of course, this paper described different barley varieties, and you can read all about it when you log into our website. Another example was tree tobacco. Here you can see some of the graphs that are daily, so every moment 3 minutes being measured, and also between the days, like here, looking at biomass, transpiration, daily transpiration rate, and so forth, and also at the soil water content. Another example, now let's talk a little bit about combining drought with other treatment. So you can combine with this system different treatments. So in this case, it's biostimulants, 2 different biostimulants versus fertilizer, and in drought and not in drought. So you can see 6 different treatments, all of them randomized on the table. The colors are expressing the randomization of the different treatments. And we will not go here through the different, the detailed analysis, but what I can share here, just as a summary, is that all the different parameters we discussed about transpiration, about stomatal conductance, biomass gain, all of those, water's efficiency, all of those are taken into account versus, of course, well irrigation, drought and recovery. And we could see very clearly, based on this profiling, which of the materials work better in which stage. And this is something that a commercial company, ICL is the biggest commercial company in Israel, they took the advantage of this study in order to implement in their marketing activities. So, and they, of course, continue to use the systems as we speak. This is another trial from ICL together with them that they allowed us to publish. You see here, again, multi-treatments, 3 levels of salinity and 3 levels of potassium, trying to understand how they influence each other, and also the potassium use efficiency. So you can see some of the analysis here, but there is many more. But again, it's the same idea, managing automatically by the system 6 different treatments within the same experiment. In this experiment, it's actually drought with a virus. This yellow leaf curl virus, which helped to promote the plant tolerance to drought. So here, the virus had a positive role. And again, this is published in our website, but this analysis was done. And I can say one thing about, in general, about detection of diseases. So detection of diseases could be very early detected on the system. And the reason is that the transpiration, the stomata activity is reacting very early to any stress to the plant. So it helps to early detect every disease and quantify also the impact for every disease. So you can see here an example of fusarium or daily transpiration. After 2-3 days already, we can start seeing the separation between the lines, and you can see that you can quantify these differences very clearly. And in the biomass, you see it in a certain delay, but you still see it very nicely also. The same for root rot, you can see the same. So also here, you see that after day 19, when these pictures are taken after day 35, without taking these roots out, it was very hard to detect that there is something going on with this plant. And already 2 weeks before, you could see it in the system data. So as a summary, we are talking about different use cases for the PlantArray system. Abiotic stress, by the way, we can today also handle flood. So this is another capability we added recently. Physiological studies, nutrients and bioag, early detection of diseases, seed treatments and how they impact the whole plant, and optimization of growth practices. I will now move to any questions if you had. I'm really thank you so much for your listening. Let's see what we have on the chat. Okay, so somebody asked, how much does it cost, the system? One second. So the system is cost is a derivative of number of course, different elements in terms of the size of the system, the pot size. So it's hard to answer this in one go. For this, it's better just to get in touch with us and we can provide you a quote. I can say that it starts with, let's say, I can give you an idea that if, for example, a Lycor equipment cost about $40,000, something like that, 50, we can already provide you several units within this, about a dozen in this price. And where Lycor measure one plant at a time and only the leaf, and here it's the whole plant. So this is just to give you an idea, and it can go up depending on the number of units you have in your wish list. Yeah. And by the way, the price will go down quite dramatically as you go up with the number of units. So it all depends on your structure and what your aim is, what pot size, etc., which sensors, how the results from this system could be translated to field experiments. So you can do you can relate to it in two different ways. One is to do the first screening in the greenhouse and then go only with successful ones to the field. This is what's happening mostly with the commercial customers we have. So they cross out those or genetic varieties or fertilizers that they don't see that they work well on the system, and they will cross them out, and then they will put it in the field, only the ones that were successful, they will continue with them. It also helps to understand and plan your field trials much better, because you come much more ready. It's much more firm data, more quantitative, more qualitative. So you come much more ready to the field, and with the fewer candidates, and only those that have more potential. You can also go the opposite way. You can come from the field as a reverse phenotyping. If you found something successful in the field, go and then have a better understanding in the greenhouse. So this is another way to use the system that some of our customers are utilizing it. Can you provide us with an attendance certificate to include in our CV? We can arrange something. Just keep in touch with us after this seminar. Can you provide also to me with an attendee? Okay. Are there any complexities to replicate drought experiment from control condition to the field? Let me see. How can we interpret macro and micro climate variables in drought experiments? So I will try to answer first the first. Are there any complexities to replicate drought experiments from control condition to the field? So of course, that in the greenhouse, it's not one-to-one as in the field. The field has a lot of, let's call it noise, that we cannot control. In a greenhouse, you can control them. In a field, you cannot. But in general, from what I know from our customers, when they do, and especially the commercial ones, when they go to the field, they already know what to aim for, and they already know how to plan their field trials much better than they did before. So if there are any complexities to replicate, maybe there are, I'm not so much aware, but just don't forget that, again, it's a different environment. So you have to take this into account. How can we interpret macro and microclimate variables in drought experiment? I'm not sure I understand this question. Because macro and micro are quite different. So I'm not sure exactly what you meant here. Maybe we can come back to it later. Please kindly give another certificate. How small can the plants be in this system? Is it applicable in Arabidopsis? Yes, we made also, some of customers made Arabidopsis trials. You can find some papers on our website with Arabidopsis. The way we deal with it, because you have to have a certain minimum weight, is that the plants need to be probably more than one. You will need to have 5 or 6 in order to gain a certain minimum weight for the system to measure it in a good form. But yes, it's possible if you do it this way. Are all the measurements based on this super sensitive scale under the pot? I don't see any sensor hanging on the leaves of the plant. Correct. The main or the key sensor is the lysimeter, the scale. And if you want, you can add more sensors. We have also an offering of more sensors that you can add on a leaf level. If you want in DVI or IR, you can do it. You can add it. Okay, I think we are more or less at the end of it. And if, I don't know, if his Professor Aminul Islam wants to explain a bit his question, you're welcome to talk, I think, at this time. There is another question. Some researchers use salt tolerance experiments to simulate drought conditions. Could these experiments be performed in this system? Definitely. You can also combine salinity conditions together with drought. As I showed before, you can combine different treatments within the same experiment. There is, by the way, Wageningen paper about salt tolerance for quinoa. You're welcome to have a look at our website. Okay, thank you. I see that you have your answers. Excellent. Okay, so I think we come to the end of the webinar. Thank you so much for listening in, and I hope you enjoyed it. Have a good day or evening, whenever you are. Thank you. Thank you.